still, you only need one F1 individual. You need to be sure that it has the parents that you think it does. So you need to check the marker genotype of that F1 individual at the markers that are flanking your target, the polymorphic markers that are flanking your targets. BC1F1, the minimum population size is 4 individuals, and you need 11 plants to have 95% probability of carrying forward at least one individual that's heterozygous at both of those loci. And if you have in marker-assisted breeding, you do a lot of things in sets of 94. 94 because the plate that we do all of our PCR reactions in is 96 wells, and 2 of those wells should be for your parents. So you have 94 wells left for your population. You might want to leave one of those wells for a negative check. You might want to use one of those wells for a reference genotype. But you've got in any case, you've got over 90 wells in the population. And if you've got a backcross, if you're trying to do marker-assisted backcrossing or marker-assisted selection in an F2 population, looking at 2 plants, looking at 2 genes, you've got, with that one plate, you've got 99% probability of recovering at least one individual that's homozygous for the favorable alleles at both of those loci. You then need you need to do DNA sampling on every individual in your population. And the biggest problem in any marker-assisted backcrossing program is keeping track of which plant provided which DNA sample, which marker data came from which DNA sample, which seed pack was harvested from that plant, and keeping track of all of those things. And the reason that it's more important in a marker-assisted selection program is you're only advancing a tiny fraction of the population each generation. In conventional breeding, we usually advance probably 20–30% of the population every generation, so we get a chance to correct any mistakes that happen, any tracking errors that happen. But in marker-assisted selection, we don't get a chance to do that. We identify the problem, and then we have to go back and repeat the previous generation. Right now, we can do a reasonably good job. 2 people working together at Papa-Sherlock can generate up to 384 DNA samples a day for $1 to $2. Basically 40 rupees to 80 rupees per DNA sample. So that's getting up to numbers that are pretty reasonable for plant breeding. 300, 400 samples per day is pretty good numbers. You can work through 1,000 population, 1,000 individuals in a week, or maybe 10 days. And that's quite good. But you can, for some methods, you don't need that much DNA, and you don't need to have that good of a DNA sample. And you can move to methods where one person can produce up to 1,000 samples a day, or between 5 and 10 rupees per DNA sample. There are now buffer solutions for DNA extraction that are available in the market, where you take your DNA extraction tools are a plastic micropipette tube, a paper punch, and a hot water bath, and a Sharpie, indelible ink pen to write the number on the plant, write the number write the sample number on the plant, write the sample number on the tube, and then throw it in the hot water bath with the buffer at the seat at the top with the buffer and the meat biscuit inside. Put it in the hot water bath for 15 minutes, and then you can pipette out an aliquot of the solution from that tube and use that in your PCR reaction. There's too little DNA for you to check on quality by any means except running a PCR reaction. But it's cheap and fast, and one person can generate 1,000 samples a day. And most breeding programs will be happy with that. You need to genotype and flanking markers. You need to genotype at least one marker. If you have and trust a perfect marker inside of your marker DNA polymorphism, inside of the gene that's controlling the QTL, and you know that it's inside of the gene that's controlling that QTL, then you can use one perfect marker. 2 on one side and 2 on the other. If you've got 2 markers on each side, then you can select for a recombination here or a recombination here. And even if you're working with a modest sized population, a couple of hundred individuals in your backcross generation or in your backcross in an F2, you can find those recombinants, at least at on one side or the other. It's very rare that you find them on both sides in one generation. And you can do selective genotyping. You can start by just genotyping at marker B and at marker D, and then the individuals that have favorable alleles there, you genotype at marker A and marker E. But that means moving a lot of DNA samples around. You have a higher probability of making a mistake in keeping track of things. And so it often ends up being more expensive to do that than to just genotype everything in all 4 markers. You want to do the foreground selection and the selection for the recombination. They're at the markers close to your peal. And then you want to do background selection, if you can afford to do that, at marker loci that are distant from your target peal. Because you want to recover the genetic background of your elite parent as quickly as possible. And there are many ways of doing this. SSR markers work quite nicely for the foreground selection, but they're pretty expensive to use for background selection. AFLPs, dart markers, SNP markers, where SNP chips are available, are all possible ways of doing background selection. And if worse comes to worse, you can actually use SSRs. But we really want to try now to exploit marker-assisted backcrossing. We need to try to reduce the number of generations that are required. We can get the entire backcrossing program to move 1 or 2 target QPLs into your elite genetic background down to a period of less than 2 years. If we use large backcross 1 F1 and large backcross 2 F1 populations. That, using those large populations, allows us to select for these recombinations at short distances flanking our target QPLs. And then we are able to take those individuals, select, do background selection and loci distant from that QPL, identify the individuals that have the favorable alleles, are heterozygous for the favorable alleles at markers B and D, but have a high percentage of recovery elsewhere of the recurrent parent genome elsewhere, and then advance those genotypes. We repeat this process. We do this in the backcross 1 F1 with about 1,000 individuals. We identify approximately 400 or 500 individuals that are heterozygous for the marker, for the donor parent alleles at the markers most closely flanking our target QPL. We then use DART or SAFLP markers to reduce that number from 300 down, or from 200 or 300. Or we use, excuse me, let's go back to the slide. So we go from 1,000 down to about 500 by identifying individuals that are heterozygous in B and D. And then we go down from 500 down to something less than 100 by identifying individuals that have a recombination between A and B, or between D and E. And then with that 100 individuals, basically 196 well plate, we do the background selection, recover individuals that have 70 or 80% recurrent parent, or much higher proportion of recurrent parent genotype than you would expect for that. For the average for that generation, you may be able to get 80 up to 85 or 90% recovery of recurrent parent in the backcross 1 itself. We then repeat the exercise by advancing 2 or 3 individuals, the backcross 2 F1 generation. Repeat the exercise on that backcross 2 F1 generation for the progeny from 2 or 3 of the selected individuals in the backcross 1. And in doing that, you then recover the recombination on the other side. either at A and B, in case you got first, you were able to get the recombination between D and E in the first generation, or between D and E if you had the recombination between A and B. You end up with a small population of backcross 2F1 individuals that you need to self, and those you then produce very long-sized backcross 2F2 populations, and are able to look at populations of 24 to 96 individuals and recover your single or multiple QTL integration homozygote with better than 95% recovery of your repeat genome. And it's taken you 5 seed generations to do this. 5 generations. Many of our crops we can do this at least under greenhouse conditions or under controlled environment conditions. We can do this in less than 2 years. However, the problem is producing those large backcross 1F1 and large backcross 2F1 generations in crops like finger millet or sorghum, where you have small flowers and it's difficult to make the hand emasculations. Even worse in chickpea or lentils. It's going to be a very serious problem to try and produce such a large backcross population. You need genetic male sterility to help you make such large populations. And so on this slide, I've got a fairly simple scheme using an example from sorghum where we have markers flanking the MS3 gene, a recessive genetic male sterility gene. We can introduce that into our really elite recurrent parent background in as little as 2 or 3 generations. 2 backcross generations and 1 generation of selfing to produce a backcross 2F2 that's segregating 1/4 of the plants are male sterile, 1/4 of the plants are male fertile homozygotes, which we identify with markers and throw away, and half of the population is heterozygous. We use those individuals as pollen donors to cross over the male sterile.